Chapter 1, we'll look at cell structure and function as a review from other biology classes you've probably taken. So with cell structure, what we're going to start out here is just talking about like a typical cell. And what you can see is things like the plasma membrane, which surrounds and encases the whole cell. It's that lipid bilayer, which helps to separate the fluid environments that are, with the intracellular fluid from the extracellular fluid. We also have other extensions of the cell surface, things like cilia, which are involved with moving material across the cell surface, or microvilli, which are involved with things like absorption. We also have other organelles here too, things like our mitochondria, which helps make ATP for the cell by converting other nutrient types. We have other things like ribosomes, which are involved with protein synthesis. We have the smooth and rough ER. So you can see the rough ER is here, smooth ER is there. Rough ER is involved with making proteins that are secreted. And then we have smooth ER, which is involved with things like lipid metabolism and detoxification of drugs. Now, nearby too, we also have the Golgi apparatus, which is involved with packaging materials and transporting those around the cell. And we can see that these secretory vesicles nearby, which are made by the Golgi apparatus. Now, in terms of just general components, we have the plasma membrane. We know this is our lipid bilayer, which is made of phospholipids, and it's primarily hydrophobic or lipophilic. And as a result, it actually separates the two fluid environments, intracellular versus extracellular fluid. And this is pretty important for cell structure because it's kind of like the skin of the cell, right? It sort of surrounds the outside and protects, all of the internal components. But we also say that it's selectively permeable because it can allow certain things to cross and other things to not cross. And we'll kind of touch back on this stuff later. Now, other cellular components, we have things like the nucleus. The nucleus contains most of the cell's genetic material. The mitochondria have some genetic material themselves, but nucleus contains basically most of your genetic material, so in the form of DNA. Within this, you have nucleoplasm, and so that's going to have like DNA and other packaging proteins. And then we have the nucleolus, which is a place where ribosomal RNA is synthesized within the nucleus itself. Other cytoplasmic organelles you'd find were like ribosomes, which are involved with protein synthesis, endoplasmic reticulum, which we talked about with smooth and rough varieties. You want to know the functions of these particular organelles, but just as a review or summary, we know that smooth ER is involved with lipid metabolism, as well as detoxification and breakdown of certain molecules within the cell. And the rough ER is involved with synthesis of protein that is secreted either into the plasma membrane or out of the cell through exocytosis. We also have the Golgi apparatus or Golgi body. This is involved with packaging and processing as well as post-translational processing of protein. So that's where you can add like sugars onto protein or add lipids onto protein. This occurs at the level of the Golgi. And then what we have then too are things like lysosomes and peroxisomes. If you guys remember, lysosomes contain hydrolytic enzymes that are involved with degradation of intracellular material, but they're also involved with breaking down damaged organelles and even apoptosis. The peroxisomes also play a role with lipid metabolism and detoxification. In fact, peroxisomes come from smooth ER. Lysosomes are actually made by the Golgi, but peroxisomes have a very similar function to smooth ER. That's why they're involved with like lipid metabolism and detoxification. It's kind of like little mobile smooth ER units. And we also have proteasomes, which is a type of organelle that's involved with breaking down, damaged protein from within the cell. Now, another type of organelle you find within the cytoplasm is the mitochondria, which is structured kind of like a cell in itself because it has its own, double membrane. It contains its own genetic material as well as its own ribosomes, and it kind of functions like its own little cell within the cell. But our cells use this mitochondria as a source of ATP, which they can use for chemical energy to do work. And then cells also have a cytoskeleton, which is made of things like microtubules and intermediate filaments and microfilaments. And what these do is they help support the cell structure. They serve as the framework for attachment of these organelles, and they help with cell mobility as well. And so the reason why we're kind of touching back on these organelles is that when cells are stressed, often what happens is that there's evidence that these organelles don't function as well, And it's also possible too that normal cellular functions can damage the cell. And we'll kind of come back to this stuff in a future chapter. So going back and talking about the plasma membrane, we said that it was a lipid bilayer. If you guys remember, it's made of phospholipids. You have the phosphate head group and fatty acid tails. And together, what it makes is essentially a double lipid membrane here, where essentially it's mostly hydrophobic. You guys can see that these hydrophobic tails or fatty acid tails kind of protrude inward. And there's things like dissolved cholesterol within the membrane as well, which is involved with membrane stability. And the phosphate head groups, which are more polar, are exposed to the fluid environment. So you have the extracellular fluid and the intracellular fluid. And you can also have embedded proteins within here too. So you have things like transport proteins, which are involved with movement of materials across the cell membrane. Often these types of transport proteins would transport molecules that are hydrophilic and wouldn't normally cross the membrane. But if you have a little channel for it, then they can pass through. Things like water can pass through these types of channels really well. Other peripheral proteins might have enzymes attached, and these can actually function as things like receptors. So if a molecule comes here and binds this peripheral protein, it might activate an enzyme and cause an intracellular signaling cascade. You also have glycolipids, which are embedded within the membrane, and glycoproteins, and these are often used for cell attachment or cell signaling. These can often have a certain shape which can help signal to other cells like immune cells, and they can help your immune system recognize self versus non-self. Now, in terms of ATP, we know that this comes from your mitochondria. It's adenosine triphosphate. We can see our adenine group here in our ribose sugar, and there are 3 phosphate groups all linked up. Now, if you guys notice here, these phosphate bonds here between each of the phosphate groups, these are high energy bonds, and the reason being is that these are pretty polar molecules, and they're electronegative. And so by being electronegative, they kind of have a tendency to repel each other. So in order for this bond to exist, it's got to contain a lot of energy. And so what's interesting then is if you break this bond, it can actually release some energy, which your cells then can use to perform work, right? And work could be things like transport of material in the cell, building and breaking down other molecules. So there's different types of work that can be kind of derived from the energy that's within these bonds. Now, what's interesting is that your body can actually recycle its entire weight in ATP every day. And the reason why this works is that when you go to break down ATP into adenosine diphosphate and you release that inorganic phosphate, essentially it can be regenerated back into ATP through metabolism, right? And there's anaerobic versus aerobic metabolism, which we'll talk about in a little bit. But essentially these can be recycled. And what's interesting is if you can kind of calculate how much you recycle each day, it ends up being about your body's weight. But this is a pretty important molecule because it is like the energy currency for our cells, and many of our enzymes use this form of chemical energy to perform work. And so in terms of metabolism, we know that there's several key types. We're going to first talk about the anaerobic metabolism. And anaerobic means it doesn't require oxygen to occur. And so anaerobic metabolism in our eukaryotic cells occurs at the level of glycolysis, and glycolysis means breakdown of sugar, and this happens in the cytoplasm of the cell. So what happens is that when your cells absorb glucose, almost immediately it's broken down through glycolysis into pyruvate and NADH. Now pyruvate is one of the breakdown products of glucose, and you get 2 pyruvate per glucose. But then you also yield a little bit of chemical energy as well in the form of what's called NADH. And this actually helps to transport or shuttle some electrons to your mitochondria to help make additional ATP. Now pyruvate is going to be oxidized further into acetyl-CoA, and then that feeds into your citric acid cycle also within your mitochondria. But what's kind of interesting though is that glycolysis, which occurs in the cytoplasm, doesn't require any oxygen, and from this you get a net 2 ATP. It ends up being about 2 ATP to break down the glucose, and you get 4 as a result. But because you have 4 minus 2, it's a net plus 2 ATP per glucose molecule via glycolysis. And so what's interesting then is that if oxygen isn't present, then your pyruvate has to be converted to lactate because the Krebs cycle won't accept pyruvate if there's not oxygen present to kind of keep these chemical reactions running. And so what ends up happening is you get lactic acid under low oxygen conditions. And so lactic acid is, we think about as being kind of a waste, but lactic acid can be actually converted back into pyruvate later. But the problem though is that when you get too much lactic acid, that can also lead to acidosis within cells and also within your bloodstream, which can cause problems. Now, if oxygen is present, then pyruvate will continue into mitochondria through the aerobic pathways, where it can be oxidized further, and you can get a lot of ATP from that pyruvate. Now, aerobic energy metabolism occurs within your mitochondria. It receives carbon-containing nutrients from elsewhere in the cell. This could be things like pyruvate from glycolysis or glycerol from fatty acids. And these can actually feed into your Krebs cycle. Now, in the presence of oxygen, what you get then is some carbon dioxide, which is a gas, some water, and some ATP. So what's interesting then is that when you're undergoing metabolism within your cells, you get gas, water, and ATP. Well, a lot of this CO2 ends up being excreted through respiration and ventilation within your lungs. Some of it can actually be lost via your kidneys as well. You can also sweat out some of this acid. And then the water that results from this metabolism also just kind of ends up elsewhere in the body, or you might urinate it out. Now, in the citric acid cycle, this occurs within your mitochondria as well as your electron transport chain. Now, carbon-containing molecules feed into your citric acid cycle, which then from that you get things like carbon dioxide. But you also get high energy carrying molecules like NADH and FADH2, which then can deliver some of these electrons to the electron transport chain. And your electron transport chain then can actually help set up a chemical gradient to help make a lot more ATP. And so what's kind of interesting is that under oxygen conditions, you can get 34 to 36 ATP molecules of molecules of ATP per glucose. And that's different than, if you remember going back to the anaerobic system where you only get 2 ATP per glucose. So you get way more ATP per glucose molecule under oxygen conditions. This is one of the reasons why we need oxygen to breathe, or to live rather. So question here says, true or false, ATP is produced without oxygen? And we know the answer can be true because of glycolysis, where this occurs in the cytoplasm and you don't need oxygen for this to continue. But you get far less ATP from glycolysis than you would aerobic metabolism in the mitochondria. Now, with cellular communication, we know that this often requires signaling molecules as well as receptors. And these receptor proteins are often embedded within the membrane of cells, where the binding site is located on the extracellular surface, and then signaling molecules can bind to the receptor site and then can lead to different types of reactions. Now, cell surface receptors can be things like G protein linked receptors, enzyme linked receptors, and then ion channel linked receptors. Now, G protein and enzyme linked receptors are pretty similar in the sense that once the ligand binds to its receptor, whether it's G protein or enzyme linked, you get an intracellular signaling cascade where different enzymes get activated and you get kind of responses within the cell as a result of these being activated. And then the ion channel linked receptors are different because when the ligand binds to the channel, or receptor rather, it opens the receptor channel, which allows ions to flow via diffusion, either into or out of the cell, depending on the concentration gradient. Now, these cell surface receptors are sensitive to hydrophilic molecules, molecules that dissolve really well in water and therefore can't cross the plasma membrane very easily. So for the cell to receive these messages, it has to have a receptor that faces the extracellular watery environment that those molecules would be dissolved within. And then intracellular receptors, those are often sensitive to lipophilic molecules that are capable of crossing the plasma membrane. Because they're lipophilic, they can get to the receptors within the cell. Things like thyroid hormone and steroid hormones have intracellular receptors because those are lipophilic and they can cross the membrane of your cell. Now, just as a summary, to talk about diffusion here, we know that diffusion is a type of transport that our cells can use to move molecules across the membrane. And we know it's a passive process, which means it doesn't require any additional ATP for this to occur. Now, diffusion is where molecules move from areas of high concentration to low concentration, and they don't require any additional input of energy. They're just going to rely on their own internal kinetic energy or movement energy to kind of move from a high to a low concentration. Now, when we talk about simple diffusion, this is where molecules can cross the plasma membrane without the use of any type of special transport mechanism or channel. They can just diffuse right across. So think about the lipophilic molecules like we just talked about, things like thyroid hormone and steroid hormones. Those can cross the membrane via simple diffusion because they're lipophilic and they can dissolve through the lipid bilayer of your cells. Other molecules, things that are hydrophilic, would require facilitated diffusion. This is where you're going to need transport proteins to help those molecules get across the plasma membrane. We have ion channels, which can actually transport ions like sodium, potassium, and chloride. And these ion channels can be leakage or gated. If they're leakage, they're always open. And so we have examples of sodium, potassium, and chloride leakage channels, which are always open within the cells. We also have gated channels, though, too. If you guys remember going back to your nervous system unit in anatomy and physiology, we talked about voltage-gated ion channels, like the voltage-gated sodium channels, which in response to a change in voltage across the membrane, that's what can get these channels to open and then allows for additional flow of ions. And then osmosis is a type of diffusion, but it's water diffusion. And so it's the same rules here in the sense that water moves from high water concentration to low water concentration. And there's another way you can think about it too, is that osmosis is a process where water can also follow solutes. So water has a tendency to get attracted to an area of higher solute concentration. So an example of this would be like if you have more sodium within your cell than outside the cell, water might have a tendency then to flow into the cell where there's more solutes or less water. So from high water concentration to low water concentration. And so this question here says, if your patient has been given an IV solution of water, what will happen to this patient's red blood cells? And this is really just a question about osmosis. So knowing how osmosis works, and if you give your patient an IV solution of water, we're saying it's just water, there's no extra solutes, then what you're essentially doing is you're delivering water to their bloodstream, which means that they're going to have less solutes in the extracellular fluid of their blood or plasma, and they're going to have more solutes inside their red blood cells. So knowing there's that concentration difference, we would know then that water would have a tendency then to flow into the red blood cells where there's more solutes, more salts, like sodium and other types of ions. And so from that knowledge, we can know then that these cells would actually enlarge or sort of swell because they're going to accumulate more water and they might even burst or lyse. This is a problem then, is that you can't just give people water in their bloodstream directly. It has to be sort of the same tonicity that your blood would normally have. And that tonicity refers to the concentration of salts or solutes that are dissolved, within your bodily fluids. And so this next question here says, true or false, an action potential is the result of potassium movement into the cell. And we know that that's false because going back to action potentials in your nervous system, we said that action potentials occur when sodium flows into the cell through the voltage-gated sodium channels. Potassium actually has a tendency to leave the cell. And the reason why sodium and potassium flow in this direction is, again, because of their concentration gradients. So if you all remember going back to these chapters in A&P, we said that sodium flows into the cell from high to low concentration because what pumps sodium out of the cell is your sodium-potassium pump. And then potassium is in a higher concentration within your cells, which means that if you allow it to flow, it's going to leave the cell from high to low concentration. Again, what sets up this gradient is your sodium-potassium pump, which uses ATP, and that's chemical energy, to do this work. Now, what's interesting then is that because these ions have a charge and they're both positive, when sodium moves into the cell, it makes this inside the cell more positive, which we call depolarization. And when potassium leaves your cell, because you're removing positive charge, that makes the inside of the cell more negative, which we call repolarization. So we'll kind of come back to this stuff in a later chapter as well. Now, for the tissues of your body, we talked about how there's 4 major tissue types. We have epithelial, connective, muscular, and nervous. The epithelial tissues are the covering and lining tissues of your body. And so some examples of this would be things like stratified squamous epithelium of your skin, or the simple squamous epithelium inside your blood vessels, or even the simple columnar epithelium along your digestive tract. And then deep to all epithelial tissues, we had connective tissue. And so the reason why all epithelia have to have an underlying connective tissue is that epithelial tissues are avascular, which means they don't have blood vessels within it. And so as a result, all epithelial tissues have to get their nutrients from an underlying connective tissue, which is going to have blood vessels going through it. And so some examples of connective tissue we talked about were things like blood in itself is actually a connective tissue, but we also have loose and dense connective tissues. Things like loose areolar helps support epithelia often, and then we have the dense connective tissues like dense regular, which is what makes up tendons. Um, Talking about muscle, we know there's three muscle types. We have smooth, cardiac, and skeletal, and we'll kind of touch back on this stuff later. But we know that cardiac muscle is only found in the heart. Smooth muscle is within the walls of hollow organs like your digestive tract and your respiratory tract and your blood vessels. And then skeletal muscle is what attaches the bones, and it's similar to cardiac muscle, but it's under voluntary control because your somatic motor system helps connect to skeletal muscle, which you can use consciously. Now, your nervous tissue is really just, that's the only type. There's only one type of nervous tissue. And within that, it's specialized because you have neurons and neuroglia, and these neurons can actually generate action potentials or essentially electrical impulses, which this tissue can use for rapid communication around your body. And the neuroglia are a type of cells that help support nervous tissue. So with the epithelial tissues, we know that they're actually classified based on the layers and the shapes of those cells. Layered arrangement of epithelia relate to the fact that if there's one layer of cells, we call it simple. If it's multiple layers, it's stratified. And if it looks like there's multiple layers, but there's only one layer, we call it pseudostratified. And so thinking about the functions of these things, we would know that if it's a simple epithelium and there's only one layer of cells, that's not going to be very tough. And so you'd find simple epithelia in places in your body that don't require a lot of physical strength. Example, this would be the inner lining of your blood vessels, where it only has to resist blood flow and pressure. There's not a whole lot of tension beyond that. Stratified epithelia you can often find within areas that experience a lot of sort of tension and physical forces. Example of this would be like your skin, which is actually a stratified squamous epithelium. And then we also know that epithelial tissues are defined based on the shape of the cells on top of that. So then we know that squamous is flattened, cuboidal means cube-shaped, and then columnar is sort of taller than they are wide, or column-shaped. And so in terms of identifying epithelial tissues, we could say simple squamous would be one layer of flattened cells, or simple cuboidal would be one layer of cuboid cells, and so on. We also have glandular epithelium, and we know that glands are things that secrete molecules, substances, that kind of stuff. And so we have two different types of glands in our body. We have exocrine versus endocrine glands. The exocrine glands are those which produce secretions that drain into ducts. And a duct is essentially a tube that drains into a space. So an example of an exocrine gland would be like the sebaceous glands of your hair follicles, which secrete sebum or oil onto your skin. And then endocrine glands are, like endo means within, right? So endocrine means secreting within. And these actually are the glands that secrete hormones in the fluids of your body, in the extracellular fluid, like interstitial fluid or your blood plasma. And these are involved with communication. Now, connective tissues would include connective tissue proper. We have loose or areolar connective tissue, adipose, which is involved with energy storage of lipids, reticular connective tissue, which actually helps make up some of the stroma of solid organs like your lungs and your liver and your spleen, your lymph nodes. And then dense connective tissues would be things like your dense regular, dense irregular. Dense regular makes up tendons and ligaments, and then dense irregular you find supporting organs that move quite a bit, like your digestive tract. And then we also have elastic connective tissue, which you find within larger elastic arteries. Other specialized connective tissues would include things like cartilage, bone, and blood. And what's kind of interesting is you think about, well, these are very different tissues, but they're all considered connective. And the reason why they're all considered connective is that they all are derived from the same cell type, which is mesenchyme. And so mesenchyme is a specialized type of cell and tissue that helps to differentiate or turn into all the types of connective tissue. In fact, all connective tissues have three properties. You have cells, fibers, and ground substance. And that's kind of the unifying sort of factor between these connective tissue types. And again, we'll come back to this later too when we talk about stem cells. So with muscle tissues, we have skeletal, cardiac, and smooth. We know skeletal muscles involved with movement of your bones, hence the name. This is under voluntary control, and it's also striated because of the sarcomeres you find within these cells. And this is basically under voluntary control, and you use this for things like breathing, for your respiratory diaphragm, as well as movement of your skeleton. Now, your cardiac muscle, you only find your heart. It's also striated, but it's under involuntary control because what controls your cardiac muscle is actually your autonomic nervous system, which is controlled by a part of your brain that you don't think about. It's under subconscious regulation. But cardiac muscle also has autorhythmic properties because there's cells within cardiac muscle tissue that can help generate action potentials on their own spontaneously. So we know that the heart actually generates its own electrical rhythm. This actually happens to be true for a lot of types of smooth muscle as well. So when you think about smooth muscle within your digestive tract, it can also generate its own action potentials, and you'd find then that your smooth muscle has these rhythmic waves of contraction which are auto-regulated as well. Now, smooth muscle is called this because it doesn't have striations like skeletal and cardiac, because smooth muscle doesn't really have sarcomeres in the same arrangement as skeletal and cardiac, so it doesn't have that sort of striated appearance to it. Now, with this question, it says, what happens to the sarcomere when myosin slides across the actin binding sites? So the question then is saying, well, does the sarcomere get longer, shorter? Does it change in length? Or does it release this enzyme called acetylcholinesterase? And we know that when sarcomeres contract, they get shorter. And what causes them to shorten is the fact that the myosin heads of the thick filament grab onto the actin binding sites of your thin filament, and it kind of helps shorten the entire unit. And that's why when your muscles contract, they get shorter, which is actually kind of like a pulling force. And in the case of skeletal muscle, it's going to pull on the bones that it's attached to. And so with nervous tissues, we have neurons and neuroglia. Remember, neurons are the sort of main communicating cells of this tissue. They're made of a soma or cell body with little neurites or extensions attached. We have things like dendrites and axons. Dendrites we think about as like the receiving end of the neuron, and axons are the output end of the neuron. And so we have different types of neurons as well: afferent, interneuron, and efferent. Afferent refers to the direction of information travel. And so afferent neurons you'd find in the peripheral nervous system, and what they do is they help transport information towards your brain and spinal cord, hence the name afferent. Interneurons are located within your brain and spinal cord in the central nervous system. Often they can also be within some of the peripheral nervous tissue as well. But interneurons are the most abundant neuron type within your body, and these are involved with a lot of processing of information because they have a lot of connections and they connect to each other, and they can form more complex processing patterns. And then efferent neurons, again, named for the direction of information flow. Efferent means going away from the central nervous system. So efferent neurons are motor neurons, which carry information from your brain and spinal cord out to muscle. So when you think about controlling the muscles of your fingers, that would be stimulated by efferent neurons of your motor system that carry information from your brain and spinal cord out to those muscles. And then neuroglia would include things like astrocytes and oligodendrocytes, microglia, and ependymal cells. If you guys remember, astrocytes help make up the blood-brain barrier, which is a nice protective coating of your capillaries of your brain to prevent harmful molecules from getting in your nervous tissue. They're also involved with potassium spatial buffering, so they help regulate the concentration of ions in the nervous tissue. And then oligodendrocytes are the ones which help form myelin, or the insulating material of axons within your central nervous system. We have the microglia, which are essentially the immune cells of your brain and spinal cord. And then the ependymal cells are a specialized type of cell with cilia on the surface that actually help make cerebrospinal fluid, which is that nourishing fluid and protective fluid of your brain and spinal cord. Now, with intercellular junctions, we know that cells need to be held together in some way. And what's kind of interesting about these cell junctions is that they all have kind of slightly different functions. So we know that we have these things called the tight junctions, which are involved with waterproofing, and they hold cells together tightly, but not as strongly as other ones. But what they're good at is forming a nice tight barrier to prevent too much water from passing between cells. Now, we also have things like adhesion belts and hemidesmosomes and desmosomes. These three are pretty similar, in terms of their structure because they're holding cells together strongly and they're kind of linked up with the internal cytoskeleton of those cells. But these ones are more for strength. So what you'd find then is that with the adhesion belt, it's going to help adhere these cells together strongly. Hemidesmosomes help cells attach to the underlying connective tissue or basement membrane. And then desmosomes are also involved with holding cells together strongly. Again, so think strength for these three. And then the last type here we have are the gap junctions. Now these ones aren't for strength so much as for communication. And what you'd find then is that gap junctions, if you look closely, there's a very narrow tube going between this. And so what gap junctions allowed for then is molecules to spread from one cell into the next. And so they're involved with communication. And so gap junctions are particularly important in your nervous system as well as your cardiac and smooth muscle tissue, because that's often how action potentials can get from one cell into the next. You also find that gap junctions are used by certain cell types to help transport nutrients from one cell into the next as well.